All right, so I'm making this video for section 4 so you guys can catch up on it. Your objectives in this section in biology are to describe the process of cellular respiration and compare cellular respiration and photosynthesis. So that first thing, or that second objective, I'm going to do right now in these notes, right? Basically, cellular respiration versus photosynthesis, they're just opposites. Everything I did in photosynthesis, I'm just going to do backwards now in cellular respiration. So all the reactions should be somewhat familiar because they really are the same thing. They're just in the reverse. So we're going to talk about 3 different parts to cellular respiration. And I'm just going to draw a picture for glycolysis here because that's step 1. Excuse me. So this is a picture that we drew in the notes, and it's important that you have this picture, right? And that's what I'm going to focus on. Otherwise, there's 2 other parts. Step 2 is the Krebs cycle, and step 3 is my electron transport. That's where some bells should be going off in your brain. So I'm going to do electron transport in respiration here. That was the first step of photosynthesis. Oh, number 2 there, it's a cycle. The Calvin cycle was part of photosynthesis, right? We're doing all the same things, just backwards, right? Because the end product of photosynthesis was to make a glucose. Guess what I start with in respiration? I start with a glucose that I'm going to essentially break in half to make a high energy 3-carbon molecule, 2 of those, right? This is exactly the opposite of the last step of our Calvin cycle last section, right? So I'm going to break down that glucose, start breaking it down. In this first step, I'm going to be able to create some ATP. So I'm going to make 2 ATP from 2 ADP molecules. But the next part of this picture, which is why I wanted to actually draw it out, is important because what just happened, this breaking down of glucose, this glycolysis, splitting the glucose, that happened in the cytoplasm. But the next steps are going to happen in my mitochondria. And my mitochondria over here, with an inner and outer membrane, right? Before I can use that mitochondria, I'm going to have to move these 3-carbon molecules across a couple membranes, which is going to be an active transport, right? That means it's going to cost me energy. So actually at this point, I'm going to have to use 2 ATP to make 2 ADP to power the transport of those 3-carbon molecules into this mitochondrial matrix, is what we call it, inner mitochondrial matrix. And it's going to cost me all the energy that I just produced, right? I may have made 2 ATP over here, but then I have to spend it right away. So obviously this is going to be worth it. There's a reason why I would do this, right? So that's a simple version of glycolysis. I make 2 ATP when I split that glucose, but then I spend 2 ATP to move 3-carbon molecules that we're going to call pyruvates later into the mitochondria. Otherwise, we got the Krebs cycle and electron transport down here. I'm not going to go through all of those in a picture just yet because we'll do that in the next video, right? But Krebs cycle, some important things that happen is I'm ultimately going to take those 3-carbon molecules and I'm going to turn them into carbon dioxides. So remember the Calvin cycle, I took carbon dioxides and I turned them into 3-carbon molecules ultimately, right? So obviously I'm going to be able to make 3 of those carbon dioxides from the 3 carbons in this pyruvate. But in the process, I'm also going to catch a little bit of energy in ATP, which is great. I'm also going to create a few other new energy molecules that I'm not going to talk about yet. So I'll just say other energy molecules here. But ultimately, through the Krebs cycle, I'm going to make 2 ATP for every, obviously it was 2 3-carbon molecules. So then ultimately I'm going to make 6 carbon dioxides per glucose. But I'm going to end up making 2 ATP in the Krebs cycle, but I already had 2 ATP from glycolysis. Had I not spent those ATP to move these 3-carbon molecules into the mitochondria, I'd still have that 2 ATP. So at this point, after the Krebs cycle, I still only have 2 usable ATP molecules, which is pretty important for later. I did a lot of work to make other energy molecules that are going to be great for electron transport. Right? That's our next step, right? Electron transport is going to take those other energy molecules and make a bunch of ATP, right? We'll draw the pictures later, but I'm going to make 34 ATP in electron transport, right? Which is way more than the 2 that I have after Krebs cycle. So this is obviously where most of the ATP from glucose comes from. That when we break down that glucose, this is where most of that ATP comes from. There is also other byproducts of electron transport. Some energy is actually going to be lost as heat. That energy is actually important because it helps keep you warm, right? If some of that usable energy wasn't lost to the system, to your cells, right, you wouldn't actually create enough heat to keep you warm enough to continue to do reactions day in, day out. So that heat is actually an important byproduct. But otherwise, in electron transport, you make 34 ATP. But one of the keys to this whole process is electron transport doesn't work without oxygen, right? Electron transport is aerobic, whereas these 2 other processes, the Krebs cycle and glycolysis, are both technically anaerobic processes. Which means they don't need oxygen to work. But here's the trick, right? If you have oxygen, then it's worth it to do these last 2 steps. If you do not have oxygen, cells should not and will not do the Krebs cycle, right? There's many different reasons to think about for that. And we'll talk about the other cell reason later. But right now, for your purposes, what you need to understand is that it really doesn't make any sense for you to spend 2 ATP up here just to get the same 2 ATP back and have to work for it at the same time. So in class, I talked to the students about how would you really consider if I told you that you could pay me $2 to do a bunch of work for me, go clean up my lab area, would you be okay with paying me $2 to go do some work for me, clean up my lab, and then I'll just give you those $2 back when it's all said and done? The answer is clearly no. Why would anybody want that deal, right? If you were told that I'd give you 34 extra dollars when it's all said and done, then yeah, totally do it, right? If oxygen's available, the cell definitely wants to do the Krebs cycle because I can't take advantage of those other energy molecules that were created here if I don't have any oxygen available, right? Without oxygen, then these energy molecules are useless. So when it's all said and done, some of this might be misleading. Make sure that you understand, right? I don't want to necessarily put in that no oxygen because aerobic means I need oxygen, right? But otherwise, I can only take advantage of these energy molecules with oxygen available. Here's the thing I need. If I don't have oxygen available, a different color, then I'm probably just going to settle for those ATP that I had from the get-go. Because if I have anaerobic conditions, it doesn't make sense to do a lot of extra stuff just to get the same amount of energy. So Krebs cycle and electron transport essentially are a package deal. You wouldn't do one without the other. You can technically do Krebs cycle without oxygen. Beer cells would never do that. So that's all we really need from this section.